Everyone, it's Chesley. Today I'll be presenting my study titled Impact of Dietary Variations on the Microbiome of Bean Beetles, a comparative study between mung beans and green peas. In this study, I investigated how diet influences the microbiome of bean beetles, Colossus brucus malculetes, raised on mung beans and green peas. By using 16S rRNA sequencing, I analyzed the microbial communities in the beetle's gut. The results revealed significant differences in microbiome composition between the two diet groups. Let's delve into the details. Bean beetles, scientifically known as Colossus brucus malculetes, are excellent models for studying dietary impacts on gut microbiomes due to their feeding habits. The composition of gut microbiome plays a crucial role in influencing host health, metabolism, and disease resistance. Previous studies in other species have shown how diet and environment can significantly shape gut microbial communities. My main research question was to determine if bean beetles fed on mung beans differ in gut microbiome composition compared to those fed on green peas. This, this question stemmed from previous findings suggesting that variations in microbiome composition can reflect biochemical environments provided by different diets. I hypothesized that the microbiomes of bean beetles would vary significantly between those fed on mung beans and those on green peas. This hypothesis was supported by findings from studies on other insects and animals where dietary changes led to marked differences in microbial diversity and composition. Understanding the dietary impact on the microbiome of bean beetles not only enhances our knowledge of insect physiology, but also contributes to broader ecological and evolutionary studies. Additionally, studies on sex differences in gut microbiota in other organisms suggest that these findings could also have implications for understanding physiological differences within species. In this study, bean beetles were divided into two groups, each exclusively fed on either mung bean or green peas. This experiment was conducted under controlled temperature and humidity conditions to ensure consistency. Samples were collected at maturity, and stringent procedures were followed to isolate and analyze the gut microbiota. The core taxa identified in gut microbiomes of bean beetles include Proteobacteria, Firmicutes, and Actinobacteria. Additionally, unique taxa associated with each diet group, such as Thermi and Armatomondetes, were also identified. Refraction analysis revealed higher microbiome richness in beetles fed mung beans compared to those fed green peas. This was further supported by greater sequence counts in the mung bean treatment. Taxonomic distribution analysis showed differences in the dominance of specific taxa between the two diet groups. For example, there was a higher abundance of Enterobacteriaceae in the gut microbiomes of beetles fed mung beans compared to green peas. The distinct microbial communities observed in bean beetles fed different diets can be attributed to the differences in nutrient composition. These findings align with similar studies in other species, highlighting the significant impact of diet on gut microbiome composition. While this study provides valuable insights, it is limited By its focus on only two legumine types, future research could expand the range of diets to include a broader array of legumes and possibly non-legumine foods to fully capture the scope of dietary influence on microbiome diversity. In conclusion, this study demonstrates the significant impact of diet on the gut microbiome composition of bean beetles. These findings have broader implications for ecological and evolutionary studies that underscore the potential of dietary manipulation as a tool for influencing insect physiology and behavior. Thank you.